I quote, the greatest threat to our planet is the belief that someone else will save it. This is a quote by the famous conservationist Robert Swan. Good evening to one and all present here. I, Andrea Joanne David, on behalf of the Natural Science Association of St. Joseph's University, welcome you all to this webinar, which is aimed at drawing your attention to the importance of conserving the threatened. I would now like to call upon my friend Kaushik to emphasize on the same.
Good evening, everybody. My name is Kaushik, and I'll be talking to you about the conserving the threatened webinar species that we have been conducting as a part of the NSA, which is actually the abbreviation of the Natural Science Association. So, conserving the threatened. Good evening, all fellow students, professors, vice chancellor, dean, and the resource person present. It is with immense pleasure that I have the opportunity to give the welcome address for this webinar, which is aptly titled as Conserving the Threatened. Today we have with us Dr. N. Marimuthu to give us a talk on a topic which is gaining popularity day by day because of what effect it has on our lives as well as the way the nature, the environment has been progressing. The topic we are here to discuss is coral reef habitat assessment based on benthic data and variables. So, this conserving the threatened webinar is a webinar series that has been put forward by the NSA, and this will be the second talk under the same. We had our first talk, which was conserving the threatened species, which is the snow leopard. And the first talk was done by Dr. Sevang Namgayal. This webinar series called Conserving the Threatened is to create awareness about the threatened species found in India. Thank you, everybody.
Now, I call upon our National Science Association coordinator, Dr. Jay Shankar, to give his address.
Thank you, Andrea. And good evening, Dr. Marimuthu, Scientist E from the Zoological Survey of India, Government of India. Uh, Thank you for accepting our invitation to address our students on conserving the threatened talk 2. This, as Kaushik mentioned, was to create the awareness about the species that are threatened in our landscape in the Indian subcontinent, and therefore involve students in towards orient them towards pursuing career and also take up research related to the field of wildlife biology, and also in general to address the general audience and also students who are enthusiastic about wildlife. This would be a platform to create awareness about species that are threatened in our country, a country that is known for good biodiversity hotspots recognized, different biogeographical zones that are there in this landscape. So keeping that in mind, sir, we had envisaged this program. And that's when we started. Coincidentally, it happened to be the top of the country over Ladakh, the Himalayan snow leopard. And now we are dipping deep sea into the waters to check into the coral ecosystems. An ecosystem that is very sensitive and vulnerable. Students, you will be listening more about that from Dr. Marimuthu. The ecosystem that is threatened. So that's what we thought will reach on to in the talk series as a talk two in the series of Conserving the Threatened. So over to you, Doctor, about this and whatever the flow is, Andrea. Next. Thank you for accepting to enlighten our students on this particular ecosystems.
Thank you, Dr. Jayashankar. Now, I call upon the president of the Natural Science Association of St. Louis University, Abhishek, to introduce our esteemed speaker for today.
Uh, Thank you, Andrea. Hello, everyone. I'm glad introducing our guest speaker today for the Conserving the Threatened Talk to Dr. Anne Marimuthu. Dr. Marimuthu is Scientist E, an officer in charge at Zoological Survey of India, Kolkata. He has done his PhD in Environmental Science, Center of Marine and Coastal Studies, Madurai Kamraj University, Madurai, Tamil Nadu, India. He has published total 33 research papers. He is an expert in systematics and spatial analysis of Sclerian corals. Now I welcome you, sir, for the virtual stage.
Sir, you have to unmute, I believe. You're not
Am I
audible.
audible now?
Yeah, yeah.
Yeah,
Yes, sir.
yeah,
Yes, sir. Please go ahead.
yeah. Thank you. My sincere thanks to Administrative Department of St. Joseph University and Head and Coordinator of Department of Zoology, my dear friend Mr. Dr. Jay Shankar, and young budding scientist for the opportunity to discuss about my view on coral reef habitats in India. I request our Mr. or Ms. Adina, can you please share that video before starting my presentation? Can you please share that video, first video?
Yes, sir. Adina will share.
Yeah, please. Could you please start the video? so much. Due to buffering issue, I think it got stuck. Meanwhile, I have shared that video. You can get it from your people. And may I share my video as a presentation? Yeah. Am I audible now?
Yes, you are.
Yeah, thank you so much. Today, actually, I'm going to deal about the topic on coral reef habitat assessment. Here, actually, we are going to discuss about the topic on biological indicators. So what are the biological indicators in and around the coral reef habitat, and what are all ecosystem services involved, and threats, either natural and anthropogenic impact involved in coral reef habitat, and how to mitigate these issues through restoration processes. These are all things we are going to discuss about that. And moreover, the carbon sequestration processes through because before going to restoration processes, we have to check which are all the species having a high capacity of carbon sequestration, like calcium carbonate, the calcification rate of reef. We are going to discuss about this. So these are all our mandates of ministry, and each and every scientist in our ZS Zoology Survey of India, we are having some mandates, primary mandates. The first mandate is systematics. We have to name this the species, and then
Sir, one
yeah,
minute. Sir,
yes,
can this be full screen, sir? Others, can you see the full screen?
No, sir. So
Okay.
can click on slideshow.
Yes. Slideshow, sir, if
yeah,
that can
yeah.
be.
Just a minute, just a minute. Is it fine now?
To me, it's
Dr. Sai? Still it is
still
fine.
in the same mode, sir. Yeah.
Yeah, just a minute.
Can there be stop sharing and then you have to reshare, sir?
Yeah,
Then
yeah,
go
yeah.
to slideshow. Yeah.
Is it okay now?
Yes, sir.
Yes, sir.
It's fine.
Oh,
Yes.
thank you, thank you. So the mandates we have explained, and this is a small statistics about habitats present in our India. I'm just talking about corals and mangroves, so the major habitats present in our marine ecosystem. Total coral cover present in our Indian subcontinent is 2,383 square kilometer area, which is 13.06% of Central Indian Ocean coral reef cover. And the total species dependent on this coral habitat is estimated about 8,766 species, which is about 8.66 percentage of total species explored from Indian subcontinent. And mangrove cover is estimated about 4,921 square kilometer area, which is 0.15 percentage of the total land area of India, according to Forest Survey of India. And 4,822 species is totally dependent on mangrove habitat, which is a 4.76 percentage of the total species explored from India. As I told you, we are going to discuss about ecological indicators. Indicators is nothing but organisms which supports that particular habitat, either in a positive manner or negative manner. Here, you can able to see the different diverse group of coral reef. This coral reef consisting of different species in Nigerian group. You can find here fire coral, which is coming under hydrozoa, and other true corals coming under anthozoa. And the reef fishes you are finding here as a positive indicator because these reef fishes acting as a grazers. It can eat all the algal particles present over the coral reef because algae is a negative indicator for the coral reef habitat. It can eat all the algal particles so that, yes, yes,
I think by
yeah,
that's by
yeah,
accident.
yeah, no issues.
Participants, kindly keep your mic on mute.
No issues, no issues. So these reef fishes can eat all the algal particle over the corals, and then it can provide the fate for further perpetuation of baby corals. So here, the reef fishes acting as a ecological indicator, positive indicator for the coral reef habitat. So many of us actually we are thinking about we have to conserve water. We should not allow the fresh water into the marine ecosystem because the populated environment we require fresh water. But this freshwater flow to the marine ecosystem is necessary nowadays because all the sediment particle is moved towards the marine ecosystem. The 50% of the sediment particle it will be trapped by the mangrove ecosystem present in the uh, coastal area, which is necessary for perpetuation of mangroves. And then remaining 50% will be captured by the seagrass habitat. And 0% will reach over to the reef slope region where coral reef present over there because corals doesn't require any small particle of sediments or nutrients for their fate. So meanwhile, it's vice versa condition. The high energy wave environment starts from the higher depth to shallow water environment. These corals can trap all the energy particle and then it can save the seagrass and mangrove habitats. So in this way, these three habitats are interlinked with each other. So ultimately, we are depending all these reef as well as other marine habitats for our food security purpose only. So here a small statistics part for benefits from this habitat. So I told about the carbon sequestration capacity of coral reef habitat and seagrass habitats are much important in this discussion. So it can sequester, coral reef can sequester carbon particle with the range of 5 to 10 gram per meter square per day. In the condition of seagrass habitat, it can sequester with the rate of 23.65 gram of carbon per meter square per day. How it can sequester? Coral reef can sequester because calcium present in the marine water and carbon dioxide present in the atmosphere, it can combine into the calcium carbonate stone in their reef forms. So in this way, it can support mankind. So moreover, the ecosystem services rate is actually from coral reefs around 3,375 billion US dollar per year. It is estimated by Kalinin. Meanwhile, seagrass also provided 1,500 million US dollar per year. So, what is the difference between corals and coral reefs? So, corals is actually it's a functional unit of coral reefs. It's a single animal which is known as a coral, and then the reef forms the total colonies is called as a coral reefs. So in corals, you can find the tentacles in above the mouth. And so you can find the tentacle number will be 6 or 6 multiplication. So the color of the colony also it depends on the associated animal zooxanthellae. So the corals, it's a sedentary animal. It cannot get the 100% of the food for their perpetuation. So the 50% of the food it came from the photosynthesis done by the zooxanthellae associated with it. So where that zooxanthellae is there, the zooxanthellae present in gastroderm layer of that coral animal, and then it can photosynthesize, and then energy particle will be transferred from one corallite to other through this mesoglia portion. So in this way, the energy particle got transferred. So these are all the morphological characters can be used for identification of the specimen. The whole animal is called as the mouth portion is called as coral light, and the external wall material, which is known as costae, and the inner wall called as a septa. And in between the correlate structure, you can find some spaces. It is called as a xenosteum. So based on their arrangement, we can identify the species up to species level. So how we can assess the corals? So this is just a simple mathematical thing only. We are going to estimate the transition points covered by individual species. So this lady is actually estimating the species cover. For an example, 25 centimeter covered by an individual species means it is actually 25 centimeter into 100 divided by the total length of the tape, which is known as the particular percentage cover of the particular species. In this way, we can estimate the cover percentage of individual species present in the benthic substrate. So how we can estimate this benthic coverage? So before that, we have to complete our diving training. Then only we can estimate this coral coverage in any particular environment. So here, Professional Association of Diving Instructors from based at Australia, they are offering these courses for maintaining a neutral buoyancy in underwater, because we are non-invaders of the sea. You can find some dangerous animal in the benthic substrate also. So if suppose I'm coming to your home, I should not disturb any environment. Then only it will be like a harmony we can maintain. So when you are enter into the water, when you are descending into the water, you have to maintain neutral buoyancy. For that, they are offering these courses around the top 20 skills that they are offering. So it starts from open water diving courses. You can go up to 15-meter depth. And then during advanced open water diving course, you can go up to 25-meter. And then third and fourth courses, emergency responder and rescue diver, it offers you how to support your buddy system, because we cannot dive alone. We have to dive with our buddy. So how to support our divers, they can teach us. And then dive master, you can go up to 40-meter depth. Here this dive master course, it will be actually for 60 days you have to attend this course. And then assistant instructor and instructor course, it can offer you can give a license for student divers up to dive master course. So these courses are available in Goa, Andaman Nicobar Islands, Lakshadweep group of islands, and then even in Chennai also they have offered. And main instructor course is offered from Bengaluru only. That people actually their head office at Bengaluru, and then they are offering courses at Havelock Island and Port Blair Island of Andaman Nicobar Islands. So now we are going to assess the morphological pattern of corals. Majorly, we are classifying into two types. One is acroporic coral and another one is non-acroporic coral. Acroporic coral is based on the presence of acron. The top polyp is it is actually it's a higher it's a greater area when compared to the lateral side. So when compared to other corals, you cannot find this apical polyp. So whatever corals are coming with this apical polyp acron, it is coming under Acropora genus. So Acropora genus, which is having 4 different classifications based on growth forms. So those corals doesn't have any branching form, which is known as digitate. And then those who are having a branching form, it is called upon Acropora coral branching. And then those coral, it looks like a table form, Acropora tabular, and then the rejuvenated form of corals known as acroporid coral encrusting form. Like this, those corals doesn't have any acron polyp. They are having 5 different classifications: massive, that is brine corals, and then submassive, pillar type coral, and then branching forms, and then foliose type of corals, and encrusting forms. Some non-sedentary corals are also there. There is a fungia species. This is a different form as a coral mushroom form, and other non-skeletonine coral. There is a hydrosova coral, coral millipora and heliopora also present in the benthic substrate. So what are all the coral associated around the coral reef habitat, which supports 37 different phyla for their perpetuation further. So porifera, sponges, sea anemone coming under Anthozoa only, hexacoralia, and soft coral. These corals actually present in sediment more environment, and jellyfish, grazing fishes, which is more important for true coral perpetuation because it eats all the algal particles, which supports like a Swachh Bharat. It can clean the environment for further perpetuation of baby coral. And then Christmas tree worm, and then nudibranchs. Here, calcareous algae I have shown here. This is actually a calcium donor for coral reef habitat. It can donate calcium for the coral reef habitat. So it's a positive indicator. Wherever you are finding this calcareous habitat, there is a fate for coral reef for perpetuation and jellyfishes. This echinodermata, you have to know about this sea cucumber, which is coming under Schedule 1, like true corals, Scleractinia, because it eats all the sediment particle over there. When monsoon times, the more sedimentation happens due to the freshwater inflow. No, that time the more sedimentation happens during the coral reef environment. So whenever the sea cucumber occurs in that environment, it eats all the sediment particles so that the mouth portion will be cleaned in the coral reef habitat so that the corals can alive further. That's what our Indian government it has put this the sea cucumber species in a scheduled act. And brittle star, feather star, these are all daughter community, sister community of this echinodermata, feather star, sea stars, and sea urchins. So recently we have documented echinodermata of Lakshadweep during a daytime, the left-hand side portion documented during the daytime, and feather stars we have documented during the nighttime because it's a nocturnal animal. You can find many species in the nighttime itself. So because morning time it can go, it will be available in the crevices of the reefs, and then it can come up during the night time for feeding purpose. So this particular brittle star we have discovered in Lakshadweep after 140 years of exploration. It is reported in Madagascar coast by Kohler in 1905. After that, after 100 years, we have explored this particular species from the Lakshadweep 2 years back. So these are all the different growth forms of corals as well as the coral associates present in our coral reef habitat. These are all the triplet code and duplicate code is an international code we are using for estimating that a particular environment for estimating the health of the ecosystem by using this triplet code. So around 10 different growth forms of corals I have shown in the left hand side portion, and some positive indicators like a calcium donor, Calamida algae and corallina algae I have given in two triplicate code, and then other associated organisms also I have given. Here, non-healthy ecosystem components also I have given here. Soft corals, as I told you, wherever sediment, more environment is there, it can perpetuate. Wherever soft corals are there, there will be not a fate for true corals. And then algal assemblage, it can lead to the coral into algal shift. And then macroalgae and turf algae, it is actually is a negative indicator for the coral reef habitats. And other abiotic factors also I have given here. So three different assessment methods are available. One is Mankato survey and another one is line intercept transect and quarter transect. The first one is a qualitative assessment survey. The second and third one is actually quantitative survey. Right now we are employing this particular second and third for the purpose of a quantitative assessment. Why Mankato survey is employed? Because if our environment is a larger amount, so what is the habitat present in that particular environment? For that purpose, we are using this Mankato survey. It is providing only qualitative assessment like 20 to 30% or 30 to 40%, like that we can estimate that particular environment. So these are all the advantages. And line intercept transect, as I told you, how to estimate the particular species cover in the total transect. So these are all the data sheets and different statistical softwares which are supporting us for estimating that benthic cover. One is the Primer versus 7. This is a biodiversity software. It's a paid version software from Ackland. We can purchase it from that company. And then POST 2.14, this is actually a free software. We can use it for a further analysis. So this is actually is a free software from Google itself. You can download it from. So we have estimated. Sorry. Some assignment we made it in 2009 to 14 when I was in National Ocean Technology with the Zoological Survey of India. We have estimated the benthic pattern of coral reef habitat around 48 sites we have selected, and we have used this primer software for easy illustration of that benthic substrate, because a 48 site have 5 different replicates, so we cannot show it in the graphical indication. Instead of that, we have used this primer software. It's a heat map. It's a shade plot. We can easily understand the how much the intensity of individual growth forms present in the benthic substrate. The left hand side portion, you can find it here: live coral, bleached coral, and dead coral with algae, rock, rubbles. Like that, we can make a variable. And the upper side portion, you can find the different regions of the Andaman Nicobar Islands where the higher intensity of the coral present over there. In the simple heat map, we can easily estimate. And then this MDS plot, which can provide the whole 48 sites intensity of a live coral cover, easily we can estimate through this simple graph. Here, the contribution of live coral cover is more towards the red color portion, that a Port Blair group of islands. So this software, which helps us to easily we can um, we can express to the layman also can easily understand from this graph. So now we are going to use this as a simple mathematical formula. How we can use this simple mathematical formula for our benthic substrate analysis. Anyone can you explain about y equal to mx plus e? What is it? So we have studied that y equal to mx plus c in class 12 actually. So this is a straight line formula. How we can use this straight line formula?
Equation of a straight line.
Yes, 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 yes. Thank you. And this straight line formula actually, my mathematical professor told me that m is a slope. You can consider this m is like our mind. When our mind is a positive, your data will be starting from 0 to 100%. If it is a negative, it will starts from 100 to 0. So it will be easy. We can easily compare two different variables. So here I have compared two variables. One is live coral cover and another one is algal assemblage. As I told you, algal assemblage is a negative indicator. So where we can compare these two variables in a negative manner. Wherever you are estimating these 2% cover, it will be like it will starts from 100 to 0% only. So the same straight line formula we are going to use for classification and mapping of this coastal benthic substrate using depth invariant index. So Lysenka is an one remote sensing analyst. He has explored one algorithm, depth invariant index. He has used this particular slope value in Ki by Kj. In that area, he has used this slope area for estimating this classification of marine features, because all the coastal features are available inside the water. Because all the coral, because what all the factors involved in estimating these features are sediments are present in the water. If you are downloading the satellite imagery, we cannot classify these features because sediments will oppose the classification. So he has used this slope value here, and then he has destructed that sedimentation impact, so that he has used this slope value for by using this for the purpose of classifying these features. So for this analysis, we should understand about first downloading the satellite image, how we can download the satellite image, and what is about what is pixel value and digital number of satellite imagery, and then what is the ground value and ground truthing, and what is the correlation and regression analysis through this y equal to mx plus c, and then ADAS imaging software. These things we should understand. Then only we can go further for this analysis. So what is pixel? So any photograph you can find if you are going through zoom in, you can find one pixel. It will have a different digital number. So each pixel have a digital number. It starts from 0 to 255. So this Lysenga, he has classified this 0 to 255 and classified based on the color pattern. He has classified different benthic features, and then he has given different color pattern for analyzing different benthic substrates. So ground truthing, what is ground truthing information? Is nothing but you have to understand about a line intercept transect, and you have to estimate the percentage of individual species. Here, standard manner you have to estimate before going to analyze through this method. You have to at least you have to select 30 different sites in particular environment. For an example, if you are selecting a Rameswaram island, you have to select some 30 different coordinates, and then you have to go to underwater and you have to estimate how much percentage cover of that particular benthic substrate, whether it is a sandy environment or coral reef environment or seagrass habitat or seaweed habitat or dead coral with algae. Like that you have to estimate. If it is like our satellite imagery, which is having a 30-meter resolution, so you have to estimate 30-meter area. What is the dominant coral reef features or sandy features or any other variables you have to estimate, and the 30 different sites, no, you have to make a first you have to get the information, and then you have to come back, and then you have to download this Landsat image from USGS site. It's a free site only. You can download that particular satellite imagery for assessing that coral reef feature. So once you are downloading that Rameswaram island, and then you have to make us through QGIS. One software is there through QGIS. You have to make a semi-automatic classification, and then it will have some bands. So what all the bands are there? 1 minute. So it will have a 7 different bands. So we are going to assess this y equal to mx plus c. Like we are going to compare two different variable. Here I'm going to compare band 2 and band 3 of that particular coordinate area. So for an example, I'm going to check based on this downloaded image. I'm going to check this coordinate area. What is the pixel value of band 2 and band 3? And I have compared. And then I got y equal to mx plus c. M is nothing but a slope. And then I'm averaging all the slope value. And then all the average value, I have put it here in this modeler, in this Lysenga equation. And then it has given this panchromatic image from our downloaded image. Now again I'm going to check our standard values are there, no, 30 different sites already we have calculated. So the same coordinate area we have to go and we have to check the file pixel value. It is having a 0 to 255 that value. So for an example, if this standard value is a coral reef habitat, means this 133 belongs to coral reef habitat. So
Hello, sir, can you hear us? I think you're on mute mode. So you have to unmute. Abhishek, can you help him, sir?
am I audible now?
Yes, sir, you are audible. So we
Okay.
want to know if the slide is changed. It's
Yeah.
still with the equation and regression analysis of
Yeah.
your paper 2017. It's still there.
Shall
I
I
just
start?
wanted to
Yeah. Shall I
confirm.
start from here?
Yes, sir. The slide is there only.
Yeah.
We are at the equation slide. Yeah. Thank
Okay. Shall I start from Landsat 5 TM?
you. Okay,
You'd
sir.
like?
As of now, the slide is with the equation and your paper that you had put, the regression analysis, that is where it's standing as of now.
Yeah, just a minute. I will check it. Sorry, I'll come back.
Okay, sir. Yes,
No,
sir. It's
yeah.
now in Landsat 5 TM.
Yes. Yes. Better I can start from here. Then only it will be fine, I think. So, shall I proceed?
Yes, sir. With the slideshow mode, sir, it is in
Yeah.
the
Yeah. Just a minute.
traditional mode. Yes.
Is it okay?
It's yet to
Still,
upload.
still it is there. Like,
No, sir, it's yet to upload. Others, can you see the slides?
No, sir. Still
No, sir.
uploading it.
oh,
No,
just
sir. Even I'm not able to see, sir. Even I'm not able to see.
Okay.
just a minute. Just a minute.
Yes, sir.
Can you able to see?
Not yet, sir.
Not
It's not yet
Not,
uploaded.
not yet. Huh?
It's not visible, sir, yet.
Still not visible. Huh?
No, sir.
Now? Can you able to see?
No, sir.
Sir, can you try joining the meeting again?
Okay, okay, okay.
Yes, sir. The slides are visible. Yes. You have to unmute, sir. You're on mute.
Shall I proceed?
Yes, sir. Please go ahead.
Sorry, friends. Now I'll start from straight line formula. How we are going to use this straight line formula for classifying features. So in physics, actually, when depth increases, due to sedimentation, you cannot classify the benthic substrate. For that, Lysenga he has explored one algorithm for classifying benthic substrates from satellite imagery. So for that, these key points we should understand. Already I explained to you. So pixel is nothing but any photograph you can find. If you are zoom in, you will find one pixel. It will have a digital number. Based on it, it starts from 0 to 255. Based on different numbers, different benthic substrates can be classified in the satellite imagery. So for ground truthing, we have to make a standard ground truthing around about 30 different sites. We have to estimate whatever ground features more than 60% is there dominating that benthic substrate. You have to consider that particular habitat for that particular point. For example, coral reef is dominating, corals are dominating 60% in particular site means you have to consider coral as that habitat. So for satellite imagery, you can download freely from a USGS site. I have downloaded Landsat 5TM. That this satellite image I downloaded from USGS site, and I have compared the green and the red band for classifying coral reef features in the benthic substrate. So after downloading that satellite imagery, I have made a semi-automatic classification through a QGIS software, and then particular coordinate. If suppose you are selecting means you will get a different file pixel value of band 1, band 2, band 3, and we are going to compare band 2 and band 3 for comparing features for getting that slope value. So here I got a reflat slope value of 1.432, and for seagrass I got a different slope value, and the sand I got a different slope value. I have averaged all this data for Lysenga equation, and I put that slope value in mathematical modeler in Lysenga equation, and then I got that satellite imagery transformed into this panchromatic image. And then I have already ground truth values are there for 30 sites, and then in that 30 sites I have selected one particular coordinate area. This is considered as a coral reef habitat, which is having a file pixel value of 133.268. So all the 133.268 wherever it is there, it is a coral reef area. Like that, we can estimate that we can easily make a classification in the panchromatic image. So based on my classification, land portion which is having a pixel value from starting from 12 to 107, and sandy region it starts from 107 to 120, like a coral reef flat region. It started from 120 to 150, and then water portion I consider as 150 to 210. So by this way, whatever ground truth value, whether it is, I need to compare because my ground truth information is it right or not, I have to validate through multivariate analysis. It is exactly matches with my satellite imagery. So the live coral pattern is more contributed towards the bison house. In satellite imagery also, I have the classified feature shows the more intensity of that orange color pattern present in the bison house. So our ground truth area is verified through satellite imagery. In this way, we can estimate our coral reef features. We can compare the data because each and every institute they are having a different mandates. This coral reef features classification is mainly carried out by ISRO-SAC. Indian Space Research Organization Satellite Application Center, and then ground truthing part is covered by our Zoology Survey of India or other biological institute like any zoological department they can do. But we have to compare all this data and then we have to verify the data with the satellite imagery. This is most important for our young budding scientists. They have to make it very clear about ground truthing point has to be validated through satellite imagery. So what are the factors and indicators present in our ecosystem, reef habitat? So here we can some natural indicators also there and anthropogenic impact factors also available. So coral bleaching is a natural phenomena where the threshold limit for coral reef to rejuvenate further is 28 degrees centigrade because it requires a constant sea surface temperature value about 28 degrees centigrade. If it is exceed 0.6 degrees centigrade, the whatever zooxanthellae present in coral reef, it will expel from corals automatically. Coral will bleach into white color portion and the coral will die. In similar way, the suspended sediment particle, it is their threshold limit is actually 27 milligram per centimeter square area per day. If it exceeds more than that, 30 milligram means automatically coral will die. So these are all natural phenomena, and wherever developmental projects it starts, the sediment particles will be gone to the mouth portion of the corals and coral will die. So Save Samudram project also it majorly affects the coral reef habitat when they have proposed, and then when they have started when pre-planning time, it has exceeded 30 milligram per centimeter square area per day. So we have proposed that it should not be exceeded near to the coral reef habitat. They have to make it in another way. And the nutrient also, silicate, phosphate, nitrate, and nitrate are major factors. It can support algal part to go further. So algal part comes under that coral reef habitat means algal shift will occur. So wherever we are making overfishing, automatically the due to nutrients eutrophication process will happens, and then finally algal part settle over the corals and coral will die. So the grazing fishes are most important thing is the positive indicator. Wherever grazes are there, coral perpetuation will occur, so that overfishing should be avoided. So, and then from a smaller portion, the baby coral also, it is having a competition between bryozoan because the bryozoan growth rate is faster rate when compared to coral, because a 2.5 centimeter growth rate of coral, it will take 10 years period for massive forms of coral. But bryozoan, it can make it within 1 month period, it can grow up to 10 centimeter to 20 centimeter. So bryozoan, when it competes with the coral, and then coral baby will die. And then algal bloom and phytoplankton bloom, it makes lesser oxygen rate in that particular environment. It will also harmful to the coral habitat. So whenever phytoplankton bloom happens, naturally these tunicates will rejuvenate. So I request I'll show this video after finalizing our presentation. Whenever phytoplankton bloom happens, immediately this tunicate will occur in that particular environment. It will live for only 3 days. and then it can swallow all the plankton, and then it can make a balance in the environment automatically. So it will manage that oxygen portion in that particular water area, dissolved oxygen in that environment, and it will support the corals for further growth. So salt bloom and then this tunicate bloom, it is actually in the Arabian Sea. You can find many blooms may happen because whenever a phytoplankton bloom happens, immediately salt and tunicate bloom happens. So recently in 2018, we found one Pyrostrema spinosum in the environment of Bangaram Island near to the Kavarati Island. And then Forest Department, they have asked us what is the reason of occurrence this particular specimen, and then we have found that the chlorophyll rate because in situ estimation of phytoplankton bloom, if it happens, it is difficult to understand. So for that, irreversibly, we have estimated that chlorophyll rate. This is also through QGAs we have estimated, and then the chlorophyll rate has estimated about more than 28 milligram per meter cube. The normal level is less than 10 milligram per meter cube for oligotrophic environment. So we have understand that due to that chlorophyll content, phytoplankton bloom happens. So wherever phytoplankton bloom happens, this tunicate will occur, and then it can swallow all the phytoplankton bloom, and then it can manage that environment for the coral reefs. So one more assignment we have received from Lakshadweep Forest Department. Lakh number of Porpita porpita is a hydrozoa specimen cast ashore in the Kavarati Island, and then they have asked us an advisory service what is the reason behind that. So the similar way we have estimated the chlorophyll rate, whether this chlorophyll rate is matched with this cast ashore, but the feeding habitat of Porpita porpita is understood about copepod species, but copepod is not at all related to phytoplankton bloom. And then phytoplankton bloom also we have checked, but the chlorophyll rate we have found about only 4.02 milligram per meter cube, which is lesser than 10 milligram per meter cube for the oligotrophic waters. So this is not the reason we found. And then we have estimated that sea surface temperature around 250 kilometer northeast portion of Kavarati Island. It is about 30.255 we have estimated through QGI software from the satellite imagery. Normal threshold limit for coral reef habitat is, as I told you, 28 degrees centigrade. See, it is about 2.255 degrees centigrade exceeded. So normally the porpita is available in oligotrophic waters, but if it happens maximum temperature, it will make induced breeding in a high number, and then it cannot swim over the water. For that, what is the reason behind the cast ashore? And then we have estimated that windrose diagram we have developed from that MedBlue software, and then it showed that from the northeast to southwest direction, we have found 20 occurrence of unusual drift, and 40 occurrence from northeast direction. So it helped that induced breeded Porpita porpita towards the Kavarati Island, and then it settled over the coastal area. And then other what all the biological indicators present in the reef habitats are this fake corals. This is also coming under Anthozoa. It is also looking like corals, but it doesn't have any calcium carbonate stone. So their growth rate also higher when compared to corals. So it also acts as a negative indicator. And then soft corals, you know very well, because it's wherever sedimentation rate over there, it can perpetuate further, and then it can compete with the corals. Calcium Halimeda algae that I already told you, this is a positive indicator. Wherever it is there, there is a good fate for coral reefs. And then Bryozoa now is the best example here. It can overgrow the baby corals here. Is a negative indicator for the coral reef habitat. So up to 1876 to 1979 report, there were only three bleaching events happened. But from 1980 to 1993, there were 60 events happened. From 2002 up to till date, 400 events happened. So wherever, if it is exceeding 28 degrees centigrade, automatically zooxanthellae will expel over there. Here you can find some corals got rejuvenated, even though some corals got bleached, because some corals having some phenotypic clades. It can it can withstand over temperature also. So there were eight different phenotypic clades of zooxanthellae are there. Whatever corals consisting of that clade, particular clade. For an example, clade A to H, clade D is having susceptible to water temperature. This whatever corals are present having clade D, which is susceptible to water temperature, and then it can withstand during the over temperature also. So coral can recover also. Here I have documented the particular species, Poretus arizoni in 2010 got bleached. In 2011, the same corals got documented with a recovery manner. So here I have shown for understanding purpose. So the bleached coral can recover further. Chronothon starfish is also a negative indicator for coral reef habitats because it eats all the coral particle. It can eat that polyps in the benthic substrate. It's an asteroidea specimen. If you are removing manually, it can understand I'm going to die. So it can expel all their larvae and then it can die. So for that, people they are injecting bile salt in situ itself, and then for the deterioration of this chronothon starfish without removing manually. So some of the gastropod snail also acting as a negative indicator. Here I have shown this is it is having a camouflage activity. So it looks like a coral. Can you able to see that gastropod snail? It looks like a coral. It is only sucking zooxanthellae present over the coral. And then it selects one particular species of coral, Poecilopora verrucosa, all over the world, and it can sucks all the zooxanthellae present over the coral inside the gastroderm layer. The muricid bioindicator. Now we are coming to coral reproduction. It is having a two different type of reproduction. One is sexual reproduction. In Indo-Pacific region, it is estimated that from December to March, the sexual reproduction is happening. It can release a massive number of eggs and sperm at the same time from the same colony, and then it can free float, and then it can the external fertilization will occur, and then planular larvae develop, and within 2 days it will settle to the suitable substrate, and then it can perpetuate. And in asexual reproduction, after perpetuation, one coral can form into two, and two forms four, four forms eight in a budding system. This is also, it is having a two different asexual reproduction is available. One is intratentacular and extratentacular budding. So how we can estimate that a particular environment with the coral reproduction rate? And this is a particular, this is important technique before proceeding to the restoration processes. Wherever coral can reproduce, that particular environment only we have to select for restoration purpose. This is the most important method for analysis. So here I am using some artificial substrate for estimating what are all the species can reproduce. So based on that, we can estimate what all the species are resilience. So these are all 1 month and 2 month and third month exposed artificial substrate, how that coral colony settle in the artificial substrate. These are all the photographs taken under microscope. So in 1 month period, you can find a single colony, and then 2 month also it's a rejuvenated form, and third month you can find the asexual manner budding system also you can find. So in here also we are having a competitive organism also you can find. Here the first four photographs, it is showing that how bryozoan community compete with the coral polyp. So from the small age itself, coral is having a problem. So whatever coral present in our Indian subcontinent, it can overgrown from this problem, and it got rejuvenated. And between the invertebrates also competition is occurring. So, what is restoration processes? Where at all restoration process is requiring? Sometime we are finding some recipient sites. For an example, some area which is having a some good ecosystem, but a further anthropogenic impact, the coral got degraded. So in that area we require transplantation from the donor site to that recipient site. We have to transplant our coral community into the rejuvenated form. Some area is having a retranslocation because some coastal industries requires that a particular environment for pipeline installation of pipeline. It can hampers the coral reef habitat. That area we have to translocate the entire colonies from that particular environment to some other environment. So before proceeding this restoration processes, we have to conduct some requisite experiments like larval check, like resilient, what all the species are there through artificial substrate installation, and then benthic chambers also. I'm having some video, but I couldn't able to upload it. But some benthic chamber analysis are there. We have to use simple other different species in that benthic chamber, how these species can accumulate calcium carbonate through calcification processes. Which species having a high calcification rate, we have to select further for restoration process. If you are selecting a non-calcification rate of non-calcification rate capacity of some particular species, means your project will be failed. So you have to select some good resilient species with a good calcification capability species. Then only we will be succeeded in the restoration processes. So some dredging activity happens in Sultanate of Oman during my earlier studies. So that time, along with the coral, they have estimated that sedimentation rate in that particular environment, it got exceeded. So that Ministry of Environment and Climate Affairs of Sultanate of Oman, they told that you have to splice out all the coral specimen present over that particular environment into some other places, because we have to conserve all these species before proceeding to that pipeline project. So we have by using underwater sizzling, and then by using underwater cements, we have translocated all the specimens into particular environment, and then we have tagged also. Translocation and transplantation is not a big deal, but after transplantation and after translocation processes, you have to check whether algal particles present over that lesion area. So wherever algal particle is there, you have to remove by using a bristles, and then you have to clean that environment for continuously for 2 months period for further perpetuation. Then only your project will be succeeded. If you are making translocation and you are not at all making a post monitoring, then algae will overgrown over the coral, and then your project will be failed. So post transplantation process is most important for this all these processes. So why, on what basis, and where we have to restoration that transplantation process has to occur. These are all these are all the area where the rejuvenated coral colony was there. Now it got degraded because of some natural or anthropogenic impacts. So these sites I have selected for recipient site from the donor site. And this site is the donor site. I'm not disturbing this entire donor site. I'm not going to collect all the specimen. I'm going to collect a single finger, single nubbin only I'm going to collect for transplantation to the recipient site. So where I have to collect that nubbin portion, the whole colony is there, and then I have to collect a small finger-like structure which is called as nubbin. So I have to cut it into pieces, and then after cutting into pieces, I'm going to make a nursery. Why I'm going to make a nursery? I'm coming from Madurai, from Tamil Nadu, and I settled at Calcutta 6 years back. I'm the new, this is the new environment for me. I have to acclimatize for some time. then only I can rejuvenate further. So all the colonies, it has to be, the nubbins has to be, make it in a nursery for 6 to 12 months. And then whatever nubbins got passed, then only you have to select that nubbin for further settlement in the benthic substrate. If you are cutting into pieces, immediately you are transplanting in that recipient site, it will be failed because you have to select the resilient specimen of that nubbin for after 6 to 12 months, then only it will be rejuvenated further. So these are all some management activities done by our Ministry of Environment and Ministry of Environment, Forest and Climate Change. Under Zoology Survey of India, we have different projects: electrolysis process, a long-term coral reef monitoring program in Lakshadweep, and permanent transect program in Andaman Nicobar Islands. In our other institutes like National Center for Sustainable Coastal Management, they are conducting sentinel sighted program and the coral reef spatial analysis through NIOT MOS program. So we have made a transplantation program in the Gulf of Kutch, and we have made a transplantation about 900 meter square area covered with the transplanted nubbins. So these are all nubbins transplanted, and then around 60% of the specimens survived in our program. These are all electrolysis nowadays under Marine Biological Regional Center from of Zoology Survey of India in Chennai. They are doing electrolysis process in Gulf of Kutch. So every 10 years, we are conducting one international program only for coral reef habitat. Is about status and protection of coral reefs in Lakshadweep environment. So I told about Sentinel Site program in National Center for Sustainable Coastal Management. They have selected 4 different islands for this program, and then they have put some data buoys for physical processes, how physical process data can help or can deteriorate coral reef habitat. So from this, when I was in NCSEM, um, I have estimated that 4 different species as a resilient species for coral regeneration processes, and then I have recommended to the Forest Department of Tamil Nadu government for further restoration purpose through this larvae purpose, and through NIOT, we have estimated that how the wave height and current speed helps the corals for perpetuation. Here we have understood that a shaded environment, how it helps the corals for biodiversity richness. And these are all major avenues for our students. These are all the institutes under Ministry of Environment, Forest and Climate Change. They are offering many programs for young budding scientists. After completing their masters, they can join as a JRF and SRF for their PhD programs. Zoology Survey of India, Botanical Survey of India, Wildlife Institute of India, NCSEM. They are offering programs under Ministry of Earth Sciences, CMLRE, NIOT, National Center for Polar and Ocean Research at Goa. They are offering many programs. Under CSR, National Oceanography, CSMCRI, they are offering many programs. Under Department of Space, Satellite Application Center of ISRO, Indian Institute of Remote Sensing at Dehradun, and IGCAR. They are offering many programs for our students. These are all major avenues for our students. Thank you. That's all about our talk. And over to our Dr. Jay for further questions. Any questions from our students?
Yes, sir. Thank you. Thank you very much, sir, for the informative presentation and also the opportunities that you said, the initiatives that are being undertaken for conserving. Over to Chandu Kishore.
Um, Hello, good evening, everyone. It has been a great webinar so far, and I thank our resource person, Dr. N. Marimuthu sir, who gave us an informative talk on the topic regarding marine ecosystem, that is coral reef habitat. Sir, everyone present in the webinar appreciates your presence with your wonderful and informative presentation. Like what are the major difference between the coral and coral reef, and how to identify the coral reef, and about the brittle star, and how to estimate the species, and few softwares and mathematic formulas for the coral analysis, and biological indicators like Grasshopper snail and Brajo, and how to restore the corals by transplantation process. Like, you have enlightened everyone present in webinar. Once again, I thank you, sir. And now the section uh, is open the questioning. Please, all of you can come up for the questioning. Either you can keep the questions in the chat box or raise your hands to unmute your mic. Please, everyone. Guys, yes, please. 1 minute. Please unmute yourself.
Good evening, sir.
First, please carry
Sir,
on.
can you hear me? Sir, so is coral reefs used for medicines to treat the diseases?
Yes, not only corals, of all pharmaceutical products, I have read one report of all pharmaceutical products, 40% of the pharmaceutical products emerge from the marine environment. Once, since corals coming under Schedule 1 species, if you would like to study about bioactive compounds from corals, you have to get a permission from Ministry of Environment for getting a specimen. And then once you are extracting that chemicals, and then you have to go further through synthesis process. Chemistry people, they might be knowing, after identification of that chemical, then you have to go through synthesis process for further development.
And next, Suraj Kumar.
In Japan also,
Good evening, sir.
sorry to bother you. In Japan also, that coral calcium carbonate they have used for uh, dental medicines actually.
Okay, sir. Thank you.
Suraj Kumar.
Hello, good evening, sir.
Good evening.
Sir, I want to know how you got interest in corals. And thank you for the session. I'm really interested and clarify many doubts. I'm from Ramanathapuram, so many times they do smuggling works like sea
Yes.
cucumber smuggled. I saw
Yes.
in news. So you say the importance and ecological role about them. So
Yes.
I will aware of that, sir. Thank you. And how you got interest in
Yeah, it's
coral,
the
sir?
most important question for all of us actually. I did my PG in biochemistry, and then when I have gone for my PhD program in Madurai Kamaraj University, I couldn't able to get my PhD in biochemistry. Then accidentally I gone to marine biology, and then my professor asked me whether you know swimming. Then I lied, I know swimming. Then after that only I learned my swimming practices, and then I got this license, open water license. Then after open water license, then I have learned advanced open water. And then after completing my PhD, then I gone to National Ocean Technology. With the support of my coordinator, I got a dive master training. Then after completing my assignment with the NIOT, then I have gone to King Abdulaziz University for a C JEDA Transect program. There I got the support for assistant instructor course. So by this way only I entered in this subject. For coral reef, actually, why I was getting that much interest, because the carbon sequestration process, how it helps in reducing carbon dioxide in the atmosphere. I think you got the point.
Okay, sir. Ramanandi, sir. Thank you, sir.
And for confiscated material also, Zoological Survey of India primary mandate is a confiscated report actually. Um, uh, um, forest department capturing scheduled animal species, and then they would like to report that person um, for a crime. So they will send to national institute like ZSI, and then we people are here for individual species, not only coral, many scheduled items Suraj told about sea cucumber. So we are also receiving many specimens. Then we have to identify up to species level based on systematics, and then we have to report to magistrate. And then magistrate, based on our report, he'll make that a further process about this thing. So these are all mandates we have to conduct in everyday assignments actually.
Thank you, sir.
And next, Vishnu Narayan.
Hello.
Yes, please carry on.
How we can calculate.
Vishnu Narayan, your voice is not much audible. Please check it once.
My question is about carbon sequestration. How can we calculate the carbon sequestration?
Yes, I can understand your question. I told about benthic chamber analysis. It is an indigenously prepared by King Abdulaziz University in Saudi Arabia. That benthic chamber offers you how species can accumulate calcium carbonate. So here the four chamber will be there. We have to install a small nubbin. What all this species nubbin has to be transplanted. We have to keep it inside that chamber, and then we have to keep it for 1 hour period in underwater environment. And then in that chamber, it will you will have a one probe and dissolved oxygen, and dissolved oxygen you can estimate the dissolved oxygen continuously, and moreover zero time, and then 60 minutes you have to take that water sample for calcification rate analysis. This is a simple alkalinity sampling only. So whatever alkalinity value you got from 0 to 60 minutes, you have to subtract into subtract subtract, and then you can get the calcification rate of that particular species for that 1 hour period. So by this way, we can estimate the calcification rate of individual species. So whatever species is having a higher calcification rate, we have to select that particular species for transplantation purpose. If you are selecting a minimum calcification rate species, then their growth rate is much slower. So your project will be failed. So we have to use that benthic chamber before proceeding to the further restoration program. I think you can get my point.
Yes, sir. My question: how we can calculate the carbon the remote sensing, sir?
A carbon sequestration is nothing but the calcium carbonate accumulation rate only.
Yeah, but not in true ground, but using the software like spatially.
Spatially,
Using
you are asking about
yeah,
spatially.
yeah, using
You
the
are asking about
satellite.
spatially. That is it's a good important question actually. For that purpose also, you have to you don't need to use that benthic chamber. You have to select some replicates. For an example, if you are selecting a Rameswaram island, I'm talking about a Park Bay environment. It is having a 30-kilometer stretch, and then randomly you have to select some points. Around 60 points you may select. Randomly you can select points, and then you can take water samples, and then you can use that value in GIS, and then like interpolation methods are there. So what is the calcification rate, easily you can estimate from the particular environment. But for individual species, you have to use benthic chamber.
Thank you, sir.
And next, Joshua, please carry on.
So, good evening. Can you hear me?
Yeah, good evening.
Sir, a lot of local communities depend on the coral reefs for survival, right?
Yes.
So, when we create or set up protected areas, how do we balance the need for conservation and their survival?
This is also yeah, you can easily compare. 1 minute. This is also it's like it's a two different variable. If you would like to conserve particular environment, then you have to make a alternative livelihood. You should not touch that environment. Vice versa. If you are using for livelihood purpose, then you will lose your habitat. So, Jacques Yves Cousteau, that quote I would like to tell you. If our grandchildren doesn't have the opportunity to see the living coral, it is an everlasting shame on our age. So, for that purpose, you should not select for a short-term benefit. For an example, under CSM CRI program, the alternative livelihood purpose, they have introduced one algal culture in Tamil Nadu coastal area, Kapophycus alversi. Many people knows about that. Once they have introduced that algae in that environment, a small portion through current, if it touches the coral reef habitat, it has made a smothering effect on the over the coral, and then coral has degraded. So we should be very careful when we are putting this livelihood purpose. We should carefully introduce some program. So livelihood purpose is the only case we have to make alternatively without touching that environment.
Sir,
And
is it possible to make small patches of protected areas over a large landscape so that the corals can also be protected and the needs of the local communities can be met? Instead of one big protected area, what if we make small patches of protected areas?
Yes, it's a good question. I would like to tell you one story. So when they have introduced that algae person, they have introduced in Gulf of Mannar only. So Gulf of Mannar Marine Biosphere Reserve, you know very well. This is the first marine biosphere reserve from Asia. So that they have banned that algae in Gulf of Mannar, and then they have given permission for Park Bay, which is very close to Gulf of Mannar. When you are crossing Pomban Pass, the left-hand side portion is Park Bay, right-hand side is in Gulf of Mannar. They have given permission for Park Bay. So algal culture happened till date in Park Bay area, but still that a small portion of the colony, it has passed through that Pomban Pass, and then it degrade that environment of Crusader Reef. Many reports says in Coral Reef Journal also many people published the data set. So when we are thinking about a long-term basis, we should be taken as marine biosphere reserve means we should not touch anything. So for that purpose only, Tamil Nadu State Forest Department they have given 3 kilometers surrounding area. There is no livelihood purpose. It should be protected, and then marine biosphere other area they can make fishing. Like that only they have given actually permission for fisher folk.
Okay, sir, that answers my question. Thank you.
Thanks a lot.
and next, Lalit Aditya, please carry on.
Good evening, sir.
Good evening, ma.
Am I audible?
Yes.
Yes, sir. So actually, I found the session really informative. And one thing I want to ask you is that if we take up serious efforts from now, serious conservation efforts from now regarding not only coral reefs, but also to mountains and the polar ice caps in the poles, can we reverse back all the effects that humankind has created?
Yeah, it's possible. It's possible, but we have to go back like a natural way. For an example, the combustion rate we need to reduce it. Everything is interlinked with each other actually. For coral regeneration, one question I have seen: can coral be genetically modified? One question from Darshan. So this answer will be beneficial for us. I told you about some phenotypic clade of zooxanthellae helps the coral to live alive even in high temperature value. They have identified 8 different clades in coral reef environment by King Abdullah University of Science and Technology. There's 8 different phenotypic clades, clade A to H, but they have identified as clade D is resilient clade for higher temperature. So like that kind of zooxanthellae modified coral colony, if we are making if we are selecting that coral species for transplantation or translocation processes, then we can regenerate. But the thing is, the growth rate of corals are very much high actually, around 10 it will take a 10 years period, 2.5 centimeter growth rate, it will take 10 years. For acropora corals it will take 1 year period only. But whenever that threshold limit if it exceeds, the temperature exceeds, acropora coral will die fastly when compared to massive forms. So we have to select massive forms, the resilient phenotypic layered consisting coral. It is most important for regeneration. It will help us, but it will take much time.
Thank you so much, sir.
And meanwhile, we have to conserve whatever wild species present over right now. For an example, from 2002 to 2005, Park Bay corals around 32 percentage mean cover we have found under MOS program. But under ZSI 2018, we have identified as it is reduced 18 percentage. It is reduced into 18 percentage. But Acropora coral got increased up to 50 percentage. So there is a fluctuation is there, but we have to conserve our wild species. Along with that, the transplantation process has to be conducted further.
Okay. And next, Sandhya D'Souza, please carry on. Uh, Sandhya, we are unable to hear you. Please check your mic once. Ma'am, please check your mic once. We are unable to hear you. Um,
Yeah, it's a good question. There any correlation between occurrence of drupala and corals? It is very difficult to say correlation between drupala because it affects only one particular species, Posilopora verrucosa. We can find, but it's a long-term project is required. At least a minimum 10 years period. We have to estimate the data set, and then we have to make more replicates spatially, and then we can come into the conclusion. Because last 4 years only I have calculated, but I couldn't get that regression equation.
Thank you for patiently answering the question, sir. If there are any more doubts, you can.
Uh, yeah, Chandu, I'll take it over from there. So thank you for patiently answering all those questions. Students were inquisitive to know more about these sensitive ecosystems. Thank you for answering them and also giving information on the initiatives that are taken for conservation. Very interestingly, one of them asked, can we go back to square one? Can we bring back all? You rightly said the natural way. There is no magic wand or some new formula to bring things. It has to be done slowly. Sir, with that, I also request you to be witness to two things that are happening from the Natural Science Association that will happen in your presence. I will hand it over to Jitu for the launch of a new forum uh, in Sir's presence. Over to Jitu.
It's my opportunity, Dr. J.
Sir, I'll share right now.
As he tries to share, yeah. Yeah, sure.
Hello. So, good evening, everyone. We, the Natural Science Association, have forums for herping and birding. And now we are launching the Josephite Forum for Marine Biology, where, uh, 1 minute, yeah, Josephite Forum for Marine Biology, a place where marine biology enthusiasts can interact, learn more, and get updates on advancements in the field of marine biology. So, we are commencing from today. Thank you.
Thank you. So as he said, sir, there are different forums that are launched because we have a large crowd of students and they're interested in different fauna. So we have one for birding. Those interested can get into birding. They do their field visits on a weekly basis and holidays and document species and come out with checklist. Similarly, to record the herpetofauna, we have a forum for the herpers. Now, there are people interested, though we are in Bangalore and landlocked. We have students from across the country, and many of them are interested to pursue their career in marine biology and also any aspects related to the marine ecosystems. So keeping that in mind, we are launching the Josephite Forum for Marine Biology in your presence. Thank
Great,
you
great.
for being
It's
here.
my opportunity.
And thank you.
And moreover,
And
I welcome this time actually I am welcoming students, those who would like to do any spatial analysis through multivariate analysis by using a different softwares. You may send as an intern for 15 days to Calcutta. I can help them.
Sure, sir. We have launched our master's program in zoology this year. There is opportunities for these interns, so we will get back to you
Sure,
in
sure.
that regard. And there's one more launch that is going to happen because we have to tell the world, like in scientific publication communication, we say publish or perish. Like that, we have to tell the world that we are here. So the Natural Science Association has different social media handles. We are launching something new now. I will hand it over to Adina. Adina, you have to unmute. Adina, you have to unmute, I believe. Only then we can hear you.
Sir, am I audible?
Yes, go ahead.
Yes.
Yes. So, hi, my name is Adina. Hope you all had a great session today. So, NSA is, spreading wide and getting into more social media contacts for you to send us more for more communications between us. So, we are launching our NSA Twitter account, which is Natural Science Association. You can share in your uh, articles and other news headlines, and we'll also be sharing the views of resource persons about the trending science topics that's going on. So it is listed here. The QR code is in the poster. You can also find the link in the chat box along with the other social media handles that we have, which is Instagram, Facebook, and also WhatsApp. Thank you.
Thank you, Adina. Over to Andrea for the final vote of thanks.
Yes, sir. Thank you. I would first like to thank Almighty God. Can you hear me, sir? Am I
Yes,
audible?
please go ahead.
Okay. I would first like to thank Almighty God for giving us this wonderful opportunity and blessing us the thirst for knowledge. I'd like to thank the management of St. Joseph's University, our Vice Chancellor, Father Reverend Dr. Victor Lobo SJ, our Registrar, Dr. Melvin Colasso, and our Dean of Life Sciences, Dr. Beatrice Sequeira, all the staff, supporting staff of St. Joseph's University, and the office bearers of the National Science Association. And I would also like to thank our speaker for today, Dr. Marimuthu, for his wonderful speech and wonderful presentation on how to conserve coral reefs. Thank you.
Thank you all. Thanks again to Dr. N. Marimuthu, Scientist E, Zoological Survey of India, for enlightening all our students' initiatives and also offering to help in future for internship or any other research related to marine ecosystems. Thank you, sir. I also want to mention the student coordinators for this event. As you saw, it's usually run by students
Great.
of the undergraduate course. The students who were in charge were Abhishek, Rooney, and Kaushik for this particular talk 2 on conservation of the threatened. The Josephite Forum for Marine Biology will be headed by two students, Kaushik and Jeetu Jute. And it's all the other office bearers of the Natural Science Association and my colleagues in the Department of Zoology, whom I should thank, the management for all the support and encouragement in organizing all these events. Sir, thank you once again for accepting our invitation. We will reach to you in future for any requirement related to research. Thank you,
Sure.
sir. Thank you,
Sure.
everybody.
Thank you, Dr. J.
Yes,
Thank you so much.
welcome, sir. Students, I believe the feedback form is posted on the chat box. Kindly fill in the details that is required for your attendance and certificate to be distributed. Yeah, as you fill in the details, you may leave the meet. Any further events